So as we start working on more problems and as we get into the part of class that's more review, it's a really good idea to have this equation and formula sheet out with you when you're working on things. Since this is exactly what will be available to you on the exam, it's just good to get familiar with it so you don't waste time looking for things when or make mistakes when the answer was on the equation sheet for you. You get this equation and formula sheet both for the multiple choice part and for the FRQ part. So whether you need it for all of it or not, it's available to you always. We already talked about the majority of the equations on here. Notice that on the right side, it explains what a lot of the variables in the equations mean. So that's a really good thing to notice as well. You don't even need to know what the variables mean because it tells you. The part that we're going to be working on today is this section that says laws of probability. So I just want you to have this out because as I explain them, I might not write them in these mathematical kinds of terms, but I want you to see how they're written on this sheet so that you can use that and figure out what that means to help you as you're doing practice problems and things like that. The other equation that we haven't done yet is Hardy-Weinberg. That's going to be part of the evolution unit. Next, other than that, I think we have covered pretty much everything on this sheet. Population growth, water potential, some of it was a while ago, but we have used everything on here except for this little corner right here. So we're talking about probability with genetics and Hardy-Weinberg with evolution, and that's the last couple that we have left. Okay, so just keep this out next to you, and as we talk about the addition rule and the multiplication rule of probability, that's what we're going to call those. You can kind of see how they're written on this equation sheet and compare that as well. So now we'll go back here. Last week, we briefly reviewed how to figure out probability in genetics with Punnett squares, which I think you're pretty familiar with. But you don't need to make a Punnett square in order to figure out probability. Punnett squares are really good if you're just working with like one trait or maybe two traits and you like doing those big 4x4 Punnett squares. But if you're doing any more than that, Punnett squares get really big and awkward really fast. And so you have to kind of mathematically calculate probabilities instead of actually drawing out the Punnett squares. So today we're going to talk about another way to determine the likelihood of certain genetic combinations using some mathematical rules. The first one is the multiplication rule. So turn for just a second and talk to somebody next to you. What are the chances of flipping two coins both heads up? What's the chance? Yeah. So you have different possibilities, right? Oh, my pen is off again. I hate that. So you could have like both heads. You could have heads, tails. You could have tails, heads. And you could have tails, tails. I'm going to fix that in a minute. And so 1 out of 4 possible combinations would be to get the first penny heads up and the second penny heads up. What are the chances of getting 3 coins all heads up? I'm going to do this while you talk about it. And tell them how you figured it out. There we go. So what are the chances here? So here, to figure this out, what are the chances of getting 1 coin to land heads up? 1/2 or 0.5, right? So for each coin, there's a 1 out of 2 chance that it'll land heads up. If we want this one and this one and this one to land heads up, what do you do with those 3 numbers? You multiply. So here's kind of the easiest way to remember which rule to use. Multiplication rule is for when you're looking for combinations of events. When you see the word and, that's probably a clue that you are going to be using the multiplication rule. The multiplication rule is what you use to find the probability of 2 independent events, meaning that one of them is not going to affect the other one. It doesn't matter what the first coin flips. That has no impact on the outcome of the second coin, right? So they're independent, unrelated events. What is the probability of them happening together. Okay, so when you see the word and in a question, what are the chances of this and this? That's a clue that you're probably going to be using the multiplication rule. Here's how this can apply to genetics. If a father has this genotype, he is heterozygous for trait R and heterozygous for trait Y, what is the probability of a sperm containing both dominant alleles? In other words, what is the probability that the sperm would contain this allele and this allele? So this is an and problem. So you do need to remember a little bit of meiosis here. Each of these genes is going to sort independently, right? The R's are going to separate into the alleles, into the gametes. The Y's are going to separate into the gametes, and one won't affect the other. So we are assuming Mendel's law of independent assortment. So these are independent events. So if we just think about the R's, what is the chance that one of his sperm cells is going to get the dominant allele? Yeah, 50% or 1 out of 2, right? Half of them will get the dominant, half will get the recessive. What is the chance that they will get the dominant Y? 1 out of 2. And so again, we want to know what are the chances they'll get the dominant R and the dominant Y, and means multiply. So just like with both tails landing heads, there's a 1 in 4 chance that this man's sperm will contain both dominant alleles. We're going to use the multiplication rule a little bit more as we go through, but any questions about this one at this point? All right. The other one that you have is the addition rule. And the addition rule is a way to figure out probability that one of 2 or more mutually exclusive events, meaning it has to be this one or this one. What are the chances that this or that will happen? Those you calculate by adding. So addition is the rule you use when you see the word or. What is the chance of this or that happening? If you want a really simple example of this, you can think of what is the chance of a penny landing heads or tails. You already know the answer to that, but you can think of it in terms of the addition rule. It's a 1 in 2 chance of landing heads, 1 in 2 chance of landing tails. The chance of it landing heads or tails is half plus half. It's 100%, right? Okay. In genetics, we often use these rules together. So here's one that I'll show you in a couple different ways, okay? What are the chances of heterozygous offspring resulting from the following cross? So if you have 2 heterozygous parents, we want to know what are the chances of having an offspring that's heterozygous. If we do this as a Punnett square, which I would recommend for a problem like this in real life because there's only one gene we're looking at here. So if we make one of the parents red, I'm just going to fill this all in the same color, and the other parent is going to be green, so you can tell where these alleles came from. You can see from this Punnett square that we have a 50% chance of getting heterozygous offspring, right? This one and this one. But the reason that there are 2 possible ways out of 4 to get a heterozygote is that they're getting the alleles from different parents, right? The one on the top got the dominant allele from the green parent. The one on the bottom got the dominant allele from the red parent. So really, we want to know, if we do this in terms of probability, we want to know a couple of things. First, we want to know what's the chance of getting a dominant allele from this parent and a recessive allele from this parent. Notice I said and there, so we're not adding, we're multiplying. So to find just this combination, there's a 1/2 probability of getting this dominant R. There's a 1/2 probability of getting that recessive R from the second parent. So there's a 1 in 4 chance of getting that exact genetic combination. With me so far? That's basically, I should have left my red and green in here. So this was the capital R. So that's the probability of getting this lower left-hand heterozygote right there. But there's another way to get a heterozygous offspring here. We could take the recessive allele from that parent and the dominant here. So the chances of getting this and this would again be 1/2 times 1/2. So there's a 1/4 chance of getting this heterozygote. And so if we want to know what are the chances of getting this combination or this combination, then we add these two together. So we have 1/4 plus 1/4 gives us the 1/2, which is the chance of getting one of those two. This problem makes the probability part look more complicated because it's actually a really simple problem you could do in a Punnett square, but it shows you the basic idea. Normally when we use probability rules, it's not always something this simple because most of you can just visualize this Punnett square or write it out real quick and figure out that answer. We more often use these probability rules with things that are too complicated to put into Punnett squares. So something more like this. We have 2 plants. It gives you their genotypes up there, and now we are looking at 3 characteristics, P, Y, and R, whatever those 3 traits are. And it's asking what fraction of the offspring from this cross is predicted to exhibit the recessive phenotypes for at least 2 of these 3 traits. Do you want to try this first, or do you want me to just walk through it? You want me to just walk through it? Okay. So the first thing, and this is helpful in a lot of probability problems, the first step that I would recommend is to look at each trait separately. Many of you are probably comfortable enough with Punnett squares that you can just visualize these Punnett squares, and you may not need to write this step out. You can maybe just think this, but for today we'll write it out just so we have it here to refer to. So for the trait, the P trait, we have 2 heterozygous parents. I need to make one of these look different. Call that one the big one. Sorry, I'm not drawing this very nicely today. Okay, so that's the inheritance just of that gene by itself. That's pretty easy. I'm just going to stay blue like that. The Y is one parent is heterozygous and the other one is homozygous recessive. So that would be like this. Let's see if I can squeeze this one in here. And the R's are the same. You have one parent that's heterozygous and the other one that's homozygous recessive. And like I said, if you need to draw these out, go for it. This part you might just be able to visualize in your head, but it is helpful to think about each trait separately first. Next here, we're going to think about the possible combinations that can answer the question. So it says we want to know what are the chances that they will be homozygous recessive, so show the recessive phenotype for at least 2. So those 2 could be, and for now I'm just going to list them and then we'll figure out probabilities. It could mean they're homozygous recessive for P and Y. It could be P and R. It could be Y and R. And it also says for at least 2, so it could be all 3. Are you with me on the logic so far? Now we're actually going to start doing some probability. This is definitely a problem where we're going to use both the multiplication and the addition rule. So here, we're going to figure out the probability that each of these could happen. And I'm not going to put those lines there. That's going to make it harder to see. Shoot. Did I have Y's first? So here, first we want to figure out what is the probability of having a homozygous recessive genotype for P and Y. So what are we going to do with those? Actually, let's do this first. So look at your Punnett squares over here. What are the chances of having a homozygous recessive offspring for the P's? 1/4. For these, you just look at the Punnett square. That's why we did this first. Just for the P's, it's 1 out of 4. And what about for the Y's? It's hard to see because it's at the bottom. But it's 2 out of 4 or 50%, so half. We want to know what's the chances of having this and this. If I said and, what do you put in the middle here? Multiply. So the chances of this combination is 1/8. That's the chance of being homozygous recessive for P and Y. So now I'm just going to go through and do that for each of these possible combinations. So the P's we said is a 1/4 chance. The chance of P and R, so R is also 1/2, so that is also a 1/8 chance. Next, we want to know what's the chance of being recessive for Y and R. So Y was a 50% chance, and R, so that one's going to be 1/4. And then the last one would be if we're recessive for all 3. So then it would be 1/4 times 1/2 times 1/2. Is that 1/16? Yes. Sorry, math brain not working. So, last step. Are you with me so far? So what this question is actually asking, these are all of the possible things that would give us at least 2 recessive traits. And it's asking, what are the chances of having this or this or this, or this, of having any of these combinations. So if we're saying or, what are we going to do with all of these fractions now? We're going to add them up. So we have 1/8 plus 1/8 plus 1/4 plus 1/16, which I just need to rewrite this because math and me are not, it's Monday. That does it. Thank you. I'm like that. Like I said, usually I'm really good with math. Today is not an easy day. So 9/16. Is my math working now? I know math can't be broken, but my math brain can. Okay, so the answer to this question, what are the chances that, or what fraction of the offspring will be recessive for at least 2? So it will have one of these combinations. That overall probability is 9/16. Questions? All right. In terms of probability and genetics, this for some people is the trickiest part, depending on how well your brain works in probabilities. There are practice problems for these linked in Classroom today, along with an answer key. We're not going to take a break this instant and have you do problems. If we have a little time at the end, you're welcome to bring some of these up. But what I would really recommend is over the next couple of days, the quiz that you're taking Thursday, since it's on all this genetic stuff, will obviously have Punnett square and probability problems on it. Do some of each of the different practice problems. There was one page I posted Friday. There are 2 of them attached today. Do at least a couple problems on each, check your answers, and then come and see me if you have questions, okay? But for today, we're gonna keep moving on and just go through a little bit more content since we don't have class tomorrow. So up to this point, we have been talking about traits that showed Mendelian inheritance. Remember, just by chance, the traits that Gregor Mendel studied showed one allele that was dominant and one that was recessive. But that's not always true. So talk for a minute about what these 3 terms mean, preferably without just reading the definitions off your notes. But if you're reading the definitions from your notes, see if you can give some examples of what those definitions mean and how all 3 of these are different. Turn and talk about that for a second. Okay. So what we've been talking about so far was complete dominance. And in all of these, the thing you really need to think about is what happens when an individual is heterozygous, okay? Because obviously if they only have dominant alleles, they show the dominant trait. If they only have recessive alleles, they show the recessive trait. The difference in all 3 of these is what happens when there's one of each. And so far what we've talked about and what Mendel saw in most of his traits was that there was complete dominance. In other words, one of the alleles totally covered up the phenotype of the other one, and you saw the exact same phenotype in the heterozygote that you did in one that was homozygous dominant. Incomplete and codominance are examples where that's not the case. Incomplete dominance is when you see something in between. That's kind of how I remember this. Incomplete dominance is in between the 2. It's kind of a blend of the phenotypes, though sometimes the blend is not exactly what you would picture, especially when we're talking color in living things. You know, if you have something that's red and something that's blue, when they blend, they don't always make purple, okay? But it's a different color that's kind of a mixture of the 2 in the heterozygote. So it's a completely different 3rd phenotype. And we'll look at some examples in just a second. In codominance, in the heterozygote, you see both of them side by side. So neither one is covered up, but they're also not blended together. For codominance, I actually think of like co-captains of a team. It's the same prefix. And I picture like 2 people up there standing side by side, both being captains of the team and leading everything and doing all the stuff together. But you actually see them both, and they're both just like they normally are, but you see them side by side. So you can look at those definitions. There are several examples here, and I think, did I print this slide for you with all these examples on there? Yeah. So you can actually write them in there. And for each of these, label it either complete dominance, codominance, or incomplete dominance. And then you can turn and talk to somebody next to you and check and see if you're on the right track, and then we'll go through them in a minute. Do you need another minute? Let's check and see how you did on these. So the first one talks about rhododendrons. The crossing of a red and white flower may yield a flower that has both red and white patches. What did you call that one? Codominance? We all agree? Yes. This one is codominance because you see the red and you see the white side by side. Could be stripes, could be spots, could be splotches and patches like this. But if you see red and you see white, that's codominance. In the Andalusian chicken, a type of chicken native to the Andalusia region of Spain, a white male and black female will often produce offspring that have blue-tinged feathers due to a lower amount of the pigment melanin. What'd you call that one? Yeah, this one, like I said, with color, the blend isn't always what we think. You normally think black and white make gray, but because of how the color shows up in an animal and what the pigment is like, it might look totally different, but blue tinged feathers obviously are not black and they're not white. You're not seeing spots or stripes or speckles. So this is a case of incomplete dominance. When certain types of long and short-furred rabbits are bred, their offspring have medium-length fur. What is that? Yeah, incomplete, medium is a blend. It's in between. You're not seeing some long fur and some short fur. And the last one, someone who inherits an A allele from one parent and a B allele from the other parent will express both proteins, resulting in an AB blood type. What is that? That is an example of codominance that we're going to talk a little more about in just a second. One interesting side note about this idea of dominance, when we talk about it like that, obviously there we were looking at the phenotypes of the entire organism. But depending on the scale that you look at this with, there are some interesting examples that show you, once again, things are always more complicated than the easy answers. Tay-Sachs disease is an example of this. It's a fatal disease because individuals are missing an enzyme that breaks down lipids, and so they accumulate lipids, fats, in their brain cells, and that eventually kills them. Only kids who have 2 copies of the Tay-Sachs allele have the disease, so they have to be homozygous to actually have Tay-Sachs, which means this seems to be a case of complete dominance when you're just looking at the health of the person, right? If you have at least one not Tay-Sachs allele, one dominant allele, you are healthy. If you have 2 copies of it, you have Tay-Sachs disease. But if you actually measure the amount of enzyme in heterozygous people, they don't have as much. It's actually at the enzymatic level, at the molecular level. This is actually incomplete dominance because people who have 2 dominant alleles make lots of this enzyme. People who have Tay-Sachs disease and have 2 recessive alleles make none of it. People who are heterozygous make an intermediate level of the enzyme. So if we're looking at a molecular level, this is actually incomplete dominance, but luckily for these people, that's enough for them to survive. So it prevents symptoms, and they don't appear to have any symptoms of that disease at all. I don't know, but I'm sure you can look it up. I mean, fairly rare because things that are fatal tend to weed themselves out of populations. But obviously, if you're heterozygous, you're okay. So that means the allele sticks around. Look it up. So that's just a little side note. As always in biology, things are always more complicated than they appear. All right, last thing today, we're going to talk just a little bit about blood type because this is a good example of codominance, and it's an example that comes up pretty frequently. So on this slide, you have a little chart here that reminds you a little bit about the genetics of blood type. So blood type is an example of a couple things. It is an example of codominance if you have an A and a B allele. It's also an example of a trait that has multiple alleles, which means within the entire human population, there aren't just 2. Like a lot of traits we've talked about so far, there's a dominant allele and a recessive allele, and that's it. In this case, there's an A allele that tells your body to attach a certain carbohydrate to a protein on your red blood cells. We can call that an A protein if we want to simplify it. There's the B allele that tells your cells to attach the B carbohydrate to this protein, so you get these B proteins. But there's also the recessive one, which we sometimes call the O allele. And you can see here it's written as a lowercase letter to represent that it's recessive. That doesn't tell your cell to make any of these carbohydrate chains on your proteins. So each of us still only has 2 copies of this allele, but there are a lot more possible combinations. The O allele is recessive to everything. So if you are type A, you can either, so if you have blood type A, that's these 2, you can either be homozygous type A and have 2 copies of the A allele, or you can be heterozygous type A, where you only have 1 A allele and a recessive, you can think of it as the O allele. That A allele still tells your cells to make the A proteins, and so the phenotype's the same. You still have type A blood. Same for type B. You can either be homozygous type B or heterozygous. Type AB and type O are the easiest because there's only 1 genotype for each. To be type AB, you have to have a gene for making A and a gene for B. And to be type O, you have to have 2 recessive alleles. Any questions just about the basic genetics of this? Because then we can think about a little bit of inheritance with this. So if a man has type A blood and he marries a type O woman, they have 3 children, 2 have type A blood and 1 has type O. What was the man's genotype? Turn and talk to somebody and see if you can figure it out. So let's check this one. I think most people either got there or really close. So the woman has type O blood. Type O can only be homozygous recessive. So she has to have this genotype. The man has type A blood. So he's got 2 possibilities here. One of his alleles has to have the letter A. But the other one could either be another A or it could be a recessive allele, right? We don't know. Based on his children, he has 2 children with type A blood. Those are pretty easy to figure out. If we fill in what we know for sure of the Punnett square, these 2 kids have type A blood. So those are type A kids. But he also had at least 1 kid with type O blood. So what does that tell you his second allele has to be? Which one? The lowercase I. Yep. He can't have this or else all of his kids would have type A blood. He has to have a recessive allele because then the other 50% of his kids would look like that and they'd have type O blood. Make sense? All right. Last one. Which parental genotypes result in children with all 4 blood types, or could, theoretically? What would the blood types of the parents be? Yeah, you need a heterozygous type A. Just in case you're wondering, this with the I's is the official way to write these genotypes. The I shows you that it's all the allele for the same trait. It's all the allele for blood type, for this ABO blood type. And then the A's and B's show the different versions of the allele. Sometimes, especially like when you learn this in 10th grade, you may have just written it like this because it makes your Punnett squares look simpler. And if you want to do Punnett squares like that, that's fine. Instead of using the I's, you definitely need to be familiar with the I notation because you might see it. But here we can get a kid that's type AB. We can get a type B kid. We can get a type A kid. And we can get a type O kid. All 4. Any questions about blood type inheritance? All right. So there's one more practice set attached. There are 2 of them attached in classroom today. So when you look at the keys, make sure you're looking at the right key. Set 2 does have one or maybe two problems that involve sex-linked traits, which we'll talk about either tomorrow or early next week. I forget. You can try them if you remember that and you want to just try them and see if you get them right. That's fine. Or you can skip those for now and come back to them later. That's totally up to you. Here's the stuff we have coming up this week. There's not going to be any new homework tonight or Wednesday night, other than more of these practice problems. There might be something Thursday. It depends if we finish it in class, and I'm just not sure yet. The things you can be working on between now and when we have class again on Wednesday. Those personal progress checks like we talked about, one should be done. If you didn't do it, at least do that one. Number 2 is due soon. Test corrections, if you want to work on those in these last few minutes. If you want to pick up stuff and bring them home, you can work on that. And then, like I said, we will have a quiz Thursday, and this is kind of our traditional genetics quiz, so it is going to have a lot of problems on it. Do some of the practice problems. This is also the parts of the book that you can kind of review to go along with all the stuff that we're working on right now. Do you have any questions? Okay, Kylie or Ashley, do you have any questions today?
I'm good.
All right. All right, if you are good, then you may sign off for the day, and we will see you again on Wednesday. And you guys just let me pause my recording thing, and then if anybody wants their tests to bring home and work on, you can.